My name is Maya, and this is my project. I came up with the idea for my research when I noticed the vasculature of some spinach leaves in my kitchen, and their structure was really similar to the blood vessels in the human body. Now, why does this matter? Well, in tissue engineering, one of the chief difficulties is trying to create substrate materials that mimic the structure of native human tissue. Now, due to convergent evolution, the structures of plant tissue are very similar to those of human tissue, which could actually solve this issue. Additionally, plant tissue is abundant, accessible, and biocompatible, all properties that would make it a great tissue engineering scaffold option. So I decided to investigate whether or not this novel material could actually serve as a scaffold in bone tissue engineering purposes. And I chose bone specifically because the porosity of these structures was very similar, and there isn't a widely agreed upon substrate for bone tissue engineering. So the first thing I had to do was actually create the scaffolds, and I did this through a process called decellularization. And through this process, the genetic and cellular material is removed from the plant tissues, leaving just a cell wall composition behind. The next thing I did was seed cells from the scaffolds and look at cell viability and proliferation using image analysis techniques. Lastly, I looked at osteogenic differentiation by culturing the cells in a bone growth media and looking for secretion of alkaline phosphatase, an enzyme that's used to produce the mineral composition of bone. And I did this as compared to a positive glass control to compare my results to other levels of osteogenic differentiation. So the first thing I did was characterize the scaffolds, and I chose all porous constructs because this is the structure that's most similar to cancellous bone. And you can see that there's a wide variety of pore geometries and pore sizes that are species dependent based on the plant. The next thing I saw was high levels of cell viability on all the constructs after 24 hours, and this indicates that the scaffolds are not cytotoxic. In the proliferation test, I saw significant levels of cell proliferation on the carrot and celery scaffolds, but the same wasn't true of the asparagus. And this indicates that cell behavior on plant scaffolds is dependent on the microtopography of the species the cells are cultured on. The next thing I looked at was cell behavior in terms of morphology. I was looking for the cells to be less circular and more spread, both of which I saw on the celery scaffold, and this is due to the larger pore size of the celery tissue. These pores allowed the cells to spread across the pores and assume this less circular and more spread morphology. Now, in bone tissue engineering, cell alignment is important because the cells need to be oriented a certain way in order for the tissue to function properly. But this is sometimes hard to do in tissue engineering. It's hard to align the cells a certain way. But what's really interesting is that the structures of the plant tissue could actually align the cells without any modification. For instance, the celery vascular bundle actually highly aligned the cells, which you can see in this directionality histogram. But not all plant structures could do this. The adjacent parenchymal tissue didn't align the cells the same way. One of the most interesting results were in the osteogenic differentiation test. After 7 days in culture, I saw that the cells on the plant scaffold actually started to behave like bone tissue, secreting the necessary enzymes to produce the bone matrix. Now, what's really interesting is that they actually did this at a comparable rate to the glass control. Now, the glass control is significantly stiffer than the plant tissue, and based on previous results, this would indicate that the glass control should have shown significantly higher levels of ALP production. But because this wasn't the case, this indicates that the microtopography of the plant tissue allowed the cells to spread and interact in such a way that the material was actually osteoinductive and helped the cells differentiate into bone tissue. So overall, what does this show? Well, this shows that plant tissue does have potential in tissue engineering. Additionally, because it's a novel material, these results set a precedent for cell behavior in plant scaffolds, so other researchers can leverage the biodiversity of the plant kingdom to look into other plant species for tissue engineering. Now, in the future, I'd like to get a more comprehensive view of how these scaffolds could perform in bone tissue engineering by doing in vivo studies with different animal models, and I could get an idea of osteointegrative and osteoconductive properties this way. Thank you so much for your time.